Hi friends, welcome back to NitVed Bioscience. Let's continue our discussion on the structure of the proteins. In the previous two classes, we had discussed about the primary structure of the protein and the secondary structure of the proteins, also the Ramachandran plot and collagen triple helix. Today we will see the tertiary structure of the protein. Various level of the folding is important for the functional property of the protein. Okay, just the polypeptide chain of the amino acid is not the functional. To make a functional protein, it has to undergo the various level of the folding. Primary structures and the secondary structures, it doesn't have the functional property, but the tertiary and the quaternary structures, it will have the functional property. So what is the primary structures? Primary structure is just the string of amino acid where the amino acids are held together by the peptide bond. Peptide bond has the CONH, and this way the long chain will takes place. So this is the peptide bond. So peptide bond which is holding the amino acid together, and it will form the primary structures. When this is the primary structures, in the secondary structures there is a formation of the hydrogen bond between the carbonyl oxygen of the one amino acids and the amide hydrogen of the another amino acid which is few amino acid away from it, and it will form the secondary structures. So secondary structures we had discussed is like alpha helix, beta pleated sheet like structures. So where hydrogen bond is present, and that hydrogen bond is formed between the carbonyl oxygen of the one amino acid and the amide hydrogen of the another amino acid which is few amino acid away in case of the alpha helix, or between the two chains in the sheet like the structures. So what I am trying to discuss here, primary structures, peptide bond is present. Secondary structure, hydrogen bond. Okay, primary structures, peptide bond, secondary structures, hydrogen bond. But here there is not involvement of the side chain. General structure of the protein, where is the hydrogen at one side carbonyl group, another side amino group, and the side chain is involved here. So while forming the primary structures, secondary structures, there is not the involvement of the side chain. Okay, only the backbone is involved here, means this is the backbone. When this long chain is formed like this, having the peptide bond, so this is known as a backbone, but there is not any involvement of the side chain. In the tertiary structures, there is an involvement of the side chain, means there is a interactions or the bond formation between the side chain as well as there is a backbone involvement. So that is the tertiary structure of the protein. Okay, are you getting? So just amino acid combined to each other, that is the primary structure where only the peptide bond is present. When the this primary structure undergo the one level of the folding, so that will form the secondary structure where the hydrogen bond is present but not the involvement of the side chain. Okay, when there is a interactions between the side chain, also the backbone, so that will form the tertiary structures, and the tertiary structure will form the more compact structure of the polypeptide chain. So this secondary structure, it is not present in the whole polypeptide chain, it is just a local folding of the few amino acid. So it can be, if there is a one polypeptide chain, so one polypeptide chain at the some point it has a helical structures, then it might have the turn or the loop, then the sheet like the structures. Okay, so single polypeptide chain, so the secondary structure, it is a localized folding of the few amino acid. Okay, then the secondary structures like the helical structures, turn or the loops or the sheet like structures, it is present in the whole polypeptide chain. So that way, so this polypeptide chain having the many secondary structures, they will come close to each other, or when there is an involvement of the side chain, and it will form the more compact globular structure or compact structures. So that is the tertiary structure of the protein. Okay. So tertiary structure, there is the involvement of the side chain. So today's our learning objective is to understand the tertiary structure of the proteins and all those interactions which is involved in the forming and stabilizing the tertiary structure of the protein. Okay. Tertiary structures can be of two types. Either it is a globular type of the structure or fibrous structure. In the globular type of the structures, so the polypeptide chain, it will have fold in a such a way that it has a globular or the compact structures, and this globular structure means the tertiary structure or the protein which is having the globular structure, these are the solubles. Fibrous proteins, two types, globular proteins and the fibrous proteins. So fibrous proteins are those where the polypeptide chains are the long strands or the elongated structures, and these are the insolubles. In the globular structures, the polypeptide chain can have the more than one secondary structure is present. Two or more secondary structures are present in the globular proteins, but in the fibrous proteins, only the single type of the secondary structure is present. Globular proteins, these are generally have the functional property. Fibrous proteins, it have the structural property. Okay. Examples of the globular proteins are enzymes. Okay. Enzymes means which is having the functional properties, and these fibrous proteins, these have the like connective tissue which is proteins. Now we will see the interactions or the bonds which are involved in the tertiary structures. Example of the tertiary structure, it is a myoglobin. So myoglobins we will discuss in the next video lectures because it is required, it is little bit lengthy and it requires a separate video. Today we will see all the interactions which is involved in stabilizing the tertiary structure of the proteins. So these are, there are mainly the four type of the interactions, like the disulfide linkage, hydrophobic interactions, then the hydrogen bond and ionic interactions. Okay. So and there is also the coordinate bonds, but these four are the primarily present. So, hydrophobic interactions, disulfide linkage, hydrogen bond and ionic interactions. First we will see the disulfide. So first is the disulfide linkage. When the thiol group of the two cysteine residues in a folded protein, so when the thiol group of the two cysteine residues, when it come close to each others, there is a formation of the this sulfur or the disulfide linkage is present here, and this is a covalent type of the bond, and this is the only covalent bond apart from the peptide bond. Peptide bond is also the covalent bond. So apart from peptide bond, this is one more covalent bond. This is the disulfide linkage which is holding the proteins together or in stabilizing the protein structures. Apart from peptide bond, this is one more covalent bond. Okay. So this is the disulfide linkage which is present in the tertiary structure of the. So the next type of the bond which is present in the tertiary structures or which is involved in the stabilizing the tertiary structure of the protein, it is also known as an ionic bond, or it is also known as salt bridges. So what happens? So when any acidic amino acid, it loses protons, so it will become the anion, and any basic amino acid, when it gains protons, it will make a cation. Okay. Anion and cations. So when these opposite charges having the anion cations, when it comes to close to each other, so there is a bond formation will take place here between the opposite charges. So that is an ionic bond, and this type of interaction present in the salt, therefore it is also known as a salt bridges. Okay. So first is the disulfide bond, second is the ionic bond. So here the ionic bond will form between these two groups. Okay, one is the anion, another is the cation, and it will form the ionic bond. Next is the hydrogen bond. So hydrogen bond. This is the third type of the bond which we are discussing, which is present in the tertiary structure of the proteins. So whenever the hydrogen atoms which is bonded to the oxygen or the nitrogen, like OH or NH2, okay, so the hydrogen atoms which is bonded to oxygen or the nitrogen in the amino acids, it can form a bond with the another nitrogen or oxygen. So this type of the interaction, it is known as a hydrogen bond. Okay, hydrogen atoms which is bonded to oxygen or the nitrogen in one amino acid can form a bond with the another nitrogen or oxygen of another amino acid. So this type of the interaction is known as a hydrogen bond, and this type of the interactions can be present between the side chain and side chain or side chain and backbone. Okay, so this is the third type of the bond, that is a hydrogen bond. The next is the hydrophobic bond. Hydrophobic bond or hydrophobic interactions. So what happens here? Let us understand with the one examples. So when we pour some oil in a water, so oil is a non-polar, water is a polar. So oil will expel the water and it will form a drop, means all the oil particles, it will come close to each other and it will expels water. So same way the amino acid with the aliphatic or aromatics non-polar side chain, it will come close to each other in the interior of the proteins and it will expel water. Okay, so all those non-polar groups, it will come close to each other in the interior of the proteins, and the interaction which is present here, it is known as a hydrophobic bond. And these hydrophobic interactions, these are generally present in the interior of the polypeptide or the protein which is formed here, and the others like the ionic or the polar groups, all these are present at the surface of the proteins and it will interact with the outside surface which is a water. Okay, so non-polar group interior means hydrophobic interactions having the between the side chains. So these are present interiors, and the polar or the ionic group are present on the exterior side. So this is about the introduction of the tertiary structure of the proteins and the all the interactions involved. So interactions are the disulfide bond, hydrogen bond, ionic interactions and the hydrophobic interactions. And what is the tertiary structure of the proteins? So tertiary structures means polypeptide chain, and please remember, this is the single polypeptide chains. In the single polypeptide chains, there can be the single type of the secondary structures or the many secondary structure is present when it will come close to each other, and there is the interactions of the side chain with the various types of the bond which we had discussed. So the overall compact structures is known as a tertiary structure of the proteins, and it can have the functional properties. So a most common example of here is the myoglobins, and that we will discuss in the next class. Thank you.